Good morning. We're going to do some MCA preparation today. So this is mostly going to be review. I would like you to write this down because writing it down is going to help you remember. Just a quick overview. We talked earlier in the year about when we studied cells, about the differences between prokaryotic and eukaryotic cells. So I always try to remember that you are made of eukaryotic cells, and so are animals, plants, and fungi. The biggest difference between the two is that eukaryotic cells, of course, have that membrane-bound nucleus, and prokaryotic cells do not. Prokaryotic cells are bacteria. So all bacteria are prokaryotic cells, and all prokaryotic cells are bacteria. None of them have a nucleus. They're much smaller and simpler in structure than eukaryotic cells. It's believed that prokaryotic cells probably evolved first and then gave rise to eukaryotic cells. One thing we haven't talked a lot about are viruses, and viruses are not really cells. They do have DNA, but they really don't have all the other cellular structures. So they're really just DNA kind of wrapped up in a fancy protein coat. And in order for viruses to replicate, they actually have to be they have to get their DNA into another cell. So unfortunately, sometimes that's our cells. So we do say that they're technically not considered living. They're technically considered non-cellular because they can't really produce on their own. That's why they're not really considered living. And you don't have to write all this down, but they are much smaller than even bacteria, plant and animal cells. But viruses mutate very rapidly. They're very good at surviving, if you will. So let's take a little bit of time to review what we know about cells and their organelles. I'll probably give you a diagram for you to fill out, but probably not today. The organelles on here, as you can see, are labeled, and there's just a few that I think are kind of really the highlights. First of all, the difference, so this is clearly an animal cell, and I know this because it's kind of roundish, and it also lacks this huge vacuole, like plant cells are made of mostly the same organelles as animal cells, but they have these huge vacuoles that take up sometimes up to 90% of the cell. So this right here, I would say, this dot in the center here, this is the nucleus, but then the other whole entire part here might be vacuole, and then of course we have all of these organelles in there too, the mitochondria and Golgi bodies and so on and so forth. So the big difference between plant cells and animal cells is plant cells have a large vacuole. Animal cells can still have vacuoles, but they're very, very large in those plant cells. And then, of course, the cell wall. Plants have that cell wall. They need that rigid cell wall because plants don't have skeletal systems for support like animals do. So they need the cell wall for that purpose. And then, of course, chloroplasts, which contain that magical substance chlorophyll that allows plants to photosynthesize. And that's one huge way in that plants are different than animal cells. So really quickly then, just the quick overview. The nucleus, and you have to know these, please write these down. Nucleus is the control center of the cell. It controls all the activities of the cell. The cell membrane controls what goes into and what goes out of the cell, and also protects the cell. This is really important. The cell membrane is really important in maintaining homeostasis because of that control. Endoplasmic reticulum, we abbreviate that sometimes the ER. We say that it's the intracellular highway. It moves things through the cell, especially proteins that are made. Golgi bodies then package proteins up. Ribosomes actually make proteins. So the rough endoplasmic reticulum has ribosomes attached to it. That's what makes it rough, and that's where proteins are made. When cells make proteins, some of the proteins are going to be exported out of the cell, but obviously some of the proteins need to be in the cell. The ribosomes that are attached to the endoplasmic reticulum make proteins that are going to be exported. Ribosomes floating in the cytoplasm actually make proteins that are going to be used inside of the cell. And mitochondria, this is the powerhouse of the cell. It's where it makes that energy in the form of ATP that's used in cellular functions. We talked a lot about the structure of the mitochondria. If we cut a mitochondria open, it has all these little folds inside. And remember what we said those were for? We said first of all, they're called cristae folds. They outline kind of the inner membrane, and they serve to increase the surface area. And whenever we increase surface area in natural things, it's to be more efficient at reactions. So because of these deep folds in the mitochondrial inner membrane, it allows it more surface area to make that ATP. So it becomes very efficient at making ATP. Lysosomes have enzymes that digest old organelles. Just like we have enzymes in our saliva, the lysosomes have enzymes inside of the cell. Cytoplasm, that's just kind of the gel-like fluid that kind of surrounds, hangs out in the cell. All the organelles are floating around in it. Vacuole we already talked about, and then centrioles were important in those cell division, mitosis and meiosis. So I want to talk a little bit more about the specific functions of the cell membrane, because we said it really controls what goes in and out, and that's a really big deal. It really is important in maintaining homeostasis. Remember, homeostasis is maintaining constant internal conditions, and all organisms have to maintain homeostasis in their bodies, including plants. But it really occurs or continues at that cellular level. So maintaining homeostasis is super important function of the cell membrane. One example of that is even carbon dioxide. So remember, we breathe in oxygen, and we don't breathe in oxygen to make our lungs happy. We breathe in oxygen because our cells need oxygen. So we take in that oxygen in those little air sacs in our lungs, the alveoli. That's where it crosses into our blood. And at the same time, carbon dioxide is going to diffuse out of our cells at that point. So oxygen and carbon dioxide can just diffuse right through the cell membrane. So it's very efficient at getting into the cell. Other things, the cell has to work a little harder getting through, and so we use something called active transport, which uses some of that ATP. So remember, if it uses energy, that's where some of that ATP that's made in the mitochondria actually gets used up. So active transport uses ATP. If we're moving very large things through the cell membrane, we use a process called endocytosis to take things into, and exocytosis to exit things from the cell. So endocytosis, if this is your cell, it literally just begins to surround these large particles of food through, and then this little piece here is going to completely surround and pinch off, and then it brings it in. Exocytosis is the opposite. We have proteins maybe that are made in here that need to be exited from the cell. So they move toward the cell membrane, and they attach to the cell membrane, and then the cell membrane opens up and kind of spits them out. Passive transport does not use energy, so oxygen and carbon dioxide moving through the membrane would move through passive transport, and actually that would be a type of passive transport called diffusion. So diffusion is when compounds move from high to low concentration. And I know I used the example earlier in the year. I dropped a little food coloring in water and we watched it diffuse through the liquid. That's diffusion. The food coloring would not coalesce back into a single drop by itself naturally. So diffusion doesn't use energy. Passive transport doesn't use any kind of energy. And remember back to where we talked about osmosis. Osmosis is movement of water through a membrane. So we put an egg in vinegar, which is a high water content, and then also corn syrup, which has a lower water content than what's inside of the egg. And we kind of saw the vinegar one, the egg got very large. It clearly took some liquid in through the membrane. Corn syrup was the opposite. It actually got very small and kind of shriveled looking because when we talk about in terms of environment, we would say the egg in vinegar was very hypertonic. It was very water loving. It took in water from the vinegar. The egg in syrup was hypotonic. The egg itself was hypotonic because it gave up water. We would say the syrup was hypertonic because it attracted water from the egg. And if we have a balance where neither one has a higher concentration than the other, then we say it's isotonic or the same. Then I want to talk a little bit about experiment design as well. We kind of are just finishing up with this, and hopefully you're done with yours. If you're not, please make sure you turn that in today. But we did a very controlled experiment with our plants. So you had to choose for your plant experiment kind of what you would be manipulating. So what you chose was the independent variable. So some of you chose things like the amount of sunlight, what type of light, the fertilizer effect on plants, and watering with different substances and all kinds of different things. So that would be your independent variable. So what you are, we sometimes say manipulated variable, but I know on the MCAs they're just going to refer to it as the independent variable. And then the dependent variable was what you are actually measuring. And so in your plant experiments, the dependent variable is of course plant growth. So we put the dependent variable on the y-axis. So plant growth, for example, was on the y-axis with the graphs that you all made from your experiment. And then the number of days that we tracked that, of course, we put on the bottom. And we actually did a controlled experiment where we had two plants. We applied the variable to one, and then there was another control that we didn't apply the variable to. And I think that's all I need to say. I feel like that's pretty fresh in your mind. There's some more MCA review we'll be doing tomorrow. But for today, let's make sure you've taken the notes on this video and then finish. There's a couple more Odysseyware through human interaction that a lot of you haven't quite finished yet. So please do those last two in our ecology unit. And I'll hopefully see you tomorrow.